Hello students, this is Dr. Ranjini, Assistant Professor in Biotechnology. Welcome to e-learning sessions in Immunology. Today we learn about immunization, types of immunization and the advantages and disadvantages of immunization. Immunization is the induction of immune response in an organism to prevent the disease, to eradicate the disease. That is called immunization. The goal of the immunization is in any individual is the prevention of the diseases. Immunization has accounted for the many advances in health uh, in the world. The immunization is classified into two types. One is active immunization and another one is passive immunization. Active immunization involves the injection or the introduction of or the administration of antigen or the foreign substance or the infectious agent or the toxin inside the host organism for active production of the immunity. Whereas passive immunization is the administration of the antibody or transferring of the antibody containing the serum or the sensitized lymphocyte cells for passive protection of the immunity or the passive protection of the infection in the recipient organism. So active immunization involves the production of the antibody, whereas passive immunization will protect the individual at that infection time. First, we'll take up passive immunization. Antibodies were first used to treat the disease in the late 19th century in the field of bacteriology. So, in the bacteriology in the 19th century, when they were using the antibodies to overcome the infection, so then we came about the passive immunization. The first success story involved diphtheria, a dangerous disease that obstructs the throat and airways in which it is causing the infection. In 1890, S. Kitasato and Emil Owen Baring immunized guinea pigs against diphtheria with heat treated blood products. Here you should put effort in understanding the technique. Here they are not injecting the whole organism which caused the diphtheria. Instead, they are taking heat treated blood products of the infected individual that has been recovered from the infection. So that heat treated blood products from the recovered individual was injected into the guinea pigs, and they, after injecting that, they collected the blood and they isolated the antibodies after repeated introduction of this heat treated the diphtheria blood products. So that was used to protect or to cure the diphtheria in the newly infected individuals. So this passive immunization will be used in the treatment of the tetanus, smallpox and bubonic plague, measles, hepatitis A, hepatitis B, rabies, very common, we all will be knowing, chicken pox. So all this will be treated by passive immunization. So passive immunization, we are transferring the antibodies or the sensitized cells inside the host organism who is suffering from the infection. This passive immunization is useful when no active immunization is available. When passive immunization is used in conjunction with the vaccination, that is the vaccine administration, the management of the specific effects of certain toxins and venoms, finally as an immunosuppressant. So they are used in combination to avoid the infection. The human antibodies are also used in curing the diseases or the infection. This preparation is that is the human immunoglobulin is derived from the alcohol fractionation of the pooled plasma. The antibody content of the immunoglobulin is almost all of the immunoglobulin IgG isotype and reflects the infection and immunization experience the donor pool. Three types of preparations are available, that is the standard gamma globulin for intramuscular use. Intramuscular use, standard immunoglobulin adapted for intravenous use, and specific immunoglobulins with high content of antibody against the antigen may available intravenous or intramuscular use. So you can inject these antibodies either by intravenous mean or by the into the muscles, so that is called intramuscular, or both would be used for bringing the resistance against the infection in case of passive immunization. Immunoglobulins should be given only for those indications where efficacy has been established. So you cannot put all the antibody which is isolated from the individual into the infected individual. So you have to check the efficacy whether it is preventing the disease or not. Although its side effects are minimal, so it may not cause the side effects, but we should be very careful in assessing the efficacy in curing the disease before administration. This immunization, passive immunization is having the disadvantages. Many disadvantages are there. Antibodies can be difficult and costly to produce. We all know. So these the this method of immunization involves the transfer of the antibodies. So you have to raise the antibody against the heat killed pathogen or the toxins. So isolation and the production of the antibodies in the organism, it is very difficult. In case of antibodies harvested from the animals, suppose you may inject that into the human being or to the animals, but if they are injected into the animals, the serious allergic reactions can be developed in the recipient. So soon after injecting these antibodies into the host organism or the to the infected individual may show the allergic reactions because the immunoglobulins are raised in the animals, may raise the allergic reaction or the hypersensitive reaction in case of the treated individual. The immunity conferred by the passive immunization is short lived. So it is a mean of you are just sending or transfusing the antibody in the individual who are suffering from the disease or the infection, and they actually clear the infection at that moment, but they'll not activate the lymphocytes to have the memory cells, due to which the immunity conferred by this passive immunization is short lived. It does not lead to the formation of long lasting memory immune cells. So these are the disadvantages of the passive immunization. Passive immunization is nothing but the transfer of the antibodies or the transfusion of the antibodies or the transfer of the challenged immune cells into the infected individual to avoid the infection at that moment. And it is short lived and may raise the allergic reaction, and the isolation and the production of the antibody is very difficult. Then coming to the active immunization, it is the induction of immunity after exposure to an antigen. Naturally, we raise the immunity against the infection. So that is the after exposure to an antigen, naturally the our body will develop the immunity. So that is called active immunization. Here antibodies are created by the recipient and may be stored permanently. Here the antigens will make the immune system to activate certain specific lymphocyte clonally multiply, and then inside the immune system they start producing the effector and the memory cells, and these effector cells remove the infection at that moment, and at the same time the memory lymphocyte retain the memory of that pathogen for the further or the subsequent infection. This artificial immunization is long lasting. So since the memory cells are stored in the body, the immunity will be there throughout the life. So this active immunization can be induced artificially, where microbe is injected into the person. So here you are taking the microbe and then you are injecting that into the individual, and you are making the immune system to develop the antibody or to activate the lymphocytes to produce the antibody. So before they are able to take in naturally. So if your body is not exposed to this antigen naturally, we are making the your body resistance against the particular infection by artificially injecting the pathogen inside the organism. So the microbe that is isolated will not be injected directly, it will be processed, is treated. It is treated so that it will not harm the infected person. Depending on the type of the disease, this technique also works with dead microbes, the part of the microbes, or treated toxins from the microbes. So you are taking the pathogen, you are treating that, you are processing that, you are attenuating that, and then you are injecting that into the organism to raise the resistance against the infection. So this may involve the technique may involve the dead microbes or the part of the microbes, or treated toxins from the microbes could be used for raising the active immunization. And when you inject that into the organism, that will raise the immunity. So the best example is the vaccination of the individual against the particular immunization. So that is the best example. So when you do the active immunization, the antibodies will be raised. So these antibodies should be very active in curing the infection as well as in remembering the pathogen for the further infection. So these antibodies should inactivate the soluble toxic proteins products of the bacteria. So the antitoxins, they act as antitoxins, and these antibodies should facilitate the phagocytosis and intracellular digestion of the bacteria. So these active antibodies which are produced by the active immunization, they should increase the rate of phagocytosis and they should act as the opsonins. So opsonins are the molecule, they rate up the process of phagocytosis. So they should act as the very strong opsonin. They should interact with the components of the serum complement to damage the bacterial membrane and hence cause bacteriolysis or to prevent the proliferation of the infectious viruses. So they should activate the other means of immunity to produce the heightened immune reaction. So newly appreciated are the antibodies that interact with the components of the bacterial surface to prevent the adhesion to the mucosal surfaces. So they act as the anti-adhesins, that means they prevent the attachment or the adsorption of the antibodies to the surface or the internal lining of the internal organs. And these antibodies cannot readily reach the intracellular sites of infection where viral replication occurs. So they are effective against many viral diseases in two ways. However, by interacting with the virus before initial intracellular penetration occurs, or by preventing locally replicating virus from disseminating from the site of entry to an important target organ, as in the spread of the polio virus from the gastrointestinal tract to the central nervous system, or of the rabies virus from the puncture wound to peripheral neural tissue. So these lymphocytes acting alone and antibody interacting with the lymphoid or the monocytic effector cells may also recognize the surface changes in the virus infected cells and destroy these infected cells. So that is the advantage of active immunization. The agent used for the active immunization is termed the antigen or also as immunogen or vaccine. It may consist of a live antigen or the attenuated viruses, like in case of measles or bacteria, BCG, or killed microbes in case of Vibrio cholerae. So we can use them as the living microorganism or attenuated microorganism, or killed microorganisms could be used to raise the active immunization. It may also be inactivated bacterial products, like in example in case of tetanus, or a specific single component of the bacteria like lipopolysaccharide. So components may be conjugated to a protein in order to produce immunogenicity in the individual in which you are injecting. Sometimes they also use recombinant DNA segment. For example, in case of hepatitis B virus, in which in this case it would be expressed in another living cells and then it will be injected into the individual. So this active immunization is advantageous over passive immunization, wherein with living organism is generally superior to the immunization with killed vaccines. And then it is inducing the long lived immunity in the organism compared to the passive immunization. So for example, multiple immunizations are recommended for polio virus. In case of the polio virus, intermittent booster doses are given in order to raise the immunity against the infection. And and the important factors involved in immunization are primarily active immunization produces protective antibody level more slowly than the incubation period of most of the infection. Must therefore be performed prior to exposure to the causative agent. That means you should inject the you should give the vaccination or the active immunization should be done before the individual naturally exposed to the pathogen. The route of immunization may be important determinant of successful vaccination. The way in which we inject the pathogen, it may be a living microbe or the attenuated or the heat killed microbe. The route in which it is entering inside the organism play an important role. Particularly if non-replicating immunogens are used, then the route of administration is very important. Immunization intranasally or by aerosols, which stimulates the mucosal immunity, often appears to be more successful than the perienteral or the injection against the viral or the bacterial challenges. So that is regarding the active immunization. So the active immunization is advantageous, long-lived, and it is activating the immune reaction. And at the same time, the route of administration of the pathogen inside the body is very important in maintaining the long-lasting immunity in the individual. And at the same time, you should inject these microbes before the natural infection occurs in the individual. Then it is very easy to activate the secondary immune reaction in the individual. So in this session, we learned what is immunization and the types of immunization, that is passive immunization and the active immunization, the differences between the passive and active immunization we learned, and the examples of the passive immunization and the active immunization we learned. And this is very important for each and every individual, because immunization play an important role in protecting us to avoid the onset of disease or the infection in the individual. I hope you understood this session. Thank you.